Hi, I'm Jared Salazar from Group 3 for the VMA of Veterinary Parasitology 412. So today I will be discussing the gastrointestinal nematodes of chicken. The materials used in the study are the following: gloves, cover slip, glass slide, test tube rock salt, distilled or tap water, plastic cups, graduated cylinder, beaker, weighing scale, faecal sample, compound microscope, and sieve. So first, let's discuss what is a flotation technique. Flotation technique use solutions which have higher specific gravity than the organisms to be floated so that the organisms rise to the top and the debris sinks to the bottom. The first procedure is the sample collection and weighing. Let's begin by collecting a fecal sample weighing precisely 3 grams. Transfer the collected sample into a designated plastic cup, ensuring accuracy in sample size. The next procedure is the saline solution preparation. To prepare the saline solution, weigh out 400 grams of rock salt. Place the measured rock salt into a beaker and then add 1 liter of distilled water to the beaker containing the rock salt. Vigorously stir the contents of the beaker until the rock salt is fully dissolved in the water. This saline solution serves as a medium for sample processing. After preparing the saline solution, the next step is to enhance sample preparation by adding a 2.5 ml of tap water to each fecal sample within the plastic cup. After that, utilize a graduated cylinder to accurately measure 47.5 ml of the prepared saline solution. The precise measurement is crucial for consistent and reliable results. Gently pour the 47.5 ml of saline solution into the plastic cup containing the 3 grams of feces. Using a tongue depressor, thoroughly mix the contents within the cup to ensure even distribution of the saline solution throughout the sample. The next step is sieving and filtration. The purpose of this is to remove the solid particles and debris. Perform sieving 3 times on the mixture. This sieving process is integral to obtaining a purified sample. Then after that, fill a test tube with an appropriate amount of flotation solution, ensuring it reaches slightly above the rim of the test tube. The flotation solution is vital for the next steps in the process. The next step is the cover slip placement. Carefully place a cover slip on top of the test tube filled with the flotation solution. Allow the setup to rest for a minimum of 20 minutes. During this time, helminths and other parasitic elements will float to the surface. After the resting period, lift off the cover slip from the test tube. Place the cover slip directly onto a clean glass slide. Proceed to examine the prepared sample using a compound microscope. This microscopic analysis is the key to identifying and studying of roundworms and other potential parasites within the fecal sample. By diligently following these comprehensive steps, the flotation technique can be effectively employed for the identification of parasitic worms and fecal samples, serving vital diagnostic and research purposes. These 5-month-old chicken aren't receiving proper deworming procedures or vitamin supplements. Neglecting deworming procedures can heighten the chicken's vulnerability to parasites, raising the likelihood of outbreaks and health problems among the caged brood of chickens. And the type of housing the chickens have is a chicken coop. In the morning, some of the chickens are outside for display, and in the evening, they are put back together in their cage. After our microscopic examination, we identified the species or genus of the parasite by comparing it to the eggs belonging to the class Nematoda. The egg we observed in our fecal examination was the species Escherichia galli. This identification was based on its similar size. Eggs have an oval shape and a relatively thick, sleek exterior shell that measures up to 77 to 94 by 43 to 55 micrometers. It is a protein-rich shell that resists drying out and remain in the environment for an extended duration. Escherichia galli, the prevalent and damaging roundworm, significantly affects domestic fowl, particularly chickens and turkeys, causing intestinal infestation. It resides in the bird's small intestine and occasionally appears in commercially sold eggs. Belonging to the Nematoda phylum, this parasitic roundworm found within the Escherichia genus mainly thrives in birds' intestines. Escherichia galli induces ascaridiasis, a condition impacting poultry digestion, resulting in poor growth, decreased egg production, digestive issues, and potential mortality, affecting the overall bird health productivity. Birds contract the infection by ingesting eggs present in contaminated food, water, or soil. These eggs are released in the bird's feces, mature in the environment, and become infective. Birds then consume these infective eggs, allowing the larvae to hatch and infest their digestive tracts. Preventing Escherichia coli infestations involves upholding excellent hygiene standards, adhering to regular deworming schedules, ensuring proper sanitation in poultry housing, and minimizing exposure to contaminated environments. Implementing these measures effectively controls the spread and impact of this parasite on poultry health and production. Escherichia coli follows a straightforward life cycle, transmitted to chickens when they ingest an impregnated egg found in feces of an infective host. Upon consumption, these worm eggs hatch in either the proventriculus or the duodenum of the chicken's digestive system. The newly hatched worms take around 28 to 30 days to develop into adults. After infection, the period before the birds begin shedding worm eggs in their feces, known as the pre-patent period, spans from 5 to 8 weeks. Numerous anthelmintic medications prove effective against Escherichia coli in poultry. Aiming to eradicate these roundworms from the bird's digestive system, some frequently employed medications are fenbendazole. This drug is capable of combating a range of parasites, including Escherichia coli. This medication interferes with the metabolism of the worms, resulting in their demise. Levamisole. This medication induces paralysis in the worms and ultimately eradicates them by impacting their nervous system. Albendazole, a versatile anthelmintic that targets diverse gastrointestinal parasites, including Escherichia coli. Piperazine, especially potent against roundworms such as Escherichia coli, inducing paralysis in the worms which are subsequently expelled by the bird. Ivermectin, though its main effectiveness lies in combating external parasites, it also demonstrates some effectiveness against specific internal parasites, including Escherichia coli. It is essential to administer this medication as per prescribed dosages and under veterinary supervision to ensure their efficacy and reduce the likelihood of resistance. Rotating various drugs categories may also be wise to prevent the rise of resistant in Escherichia coli populations.